Hi, my name is Geraldine van der Roha, lead developer advocate at Seqera, the company that makes Nextflow, the popular open source pipelining framework. In this talk, I'm going to tell you about some exciting new tooling that dramatically improves the Nextflow developer experience. But first, a little bit about us, because you've probably heard of Nextflow before, but you might not have heard of Seqera. As I said, we're the company that makes Nextflow. And yes, even though we're a company, Nextflow is completely free and open source. It's one of several open source software packages we produce as part of our mission to advance science for everyone through software. We also operate free services to provide resources to the scientific community, like containers and pipelines. And we build and operate infrastructure for doing science at scale, like Seqera Platform. So let's talk about Nextflow and also a little bit about NF Core, a really cool community project that has grown around Nextflow. And I'll start with the big picture question. Why do we need Nextflow? What problems does it solve? In a nutshell, modern data analysis is a lot of work. Hopefully, this is not a controversial statement. We all know the rise of high throughput data generation technologies has produced not only tremendous opportunity for scientific discovery, but also commensurate challenges in terms of managing and analyzing these enormous piles of data. The analysis process, in particular, typically involves putting the data through multiple steps that use a variety of different command line tools. And of course, a lot of them are special snowflakes with their own special compute requirements. And yet, the processing needs to be fully reproducible and as automated as possible. And that is why we formalize these analyses in the form of scripted workflows that we can run as automated data processing pipelines on high performance computing infrastructure. Here, to illustrate, I'm showing you a pipeline for RNA-seq analysis from the NF Core project. The specifics don't really matter. The point is that we're able to describe the entire analysis process in a machine-readable way. Of course, Nextflow isn't the only pipelining framework out there, so let's talk about some of the requirements a good pipelining framework should satisfy. Generally speaking, you're going to want a solution that can handle very large files. For example, whole genome sequencing routinely produces files above 100 gigabytes for a single sample. You also need it to be able to wrap tools and scripts written in a variety of different languages, because at any given step in the pipeline, you might need to run a Java tool, a Python package, or compile binary written in C, for example. And you need to be able to describe potentially complex graphs with paths branching and joining, sometimes determining dynamically what needs to be done to the data, depending on the outcome of an earlier step. And finally, you want to use a framework that scales with the maturity of your project. Many projects start out as a prototype in the hands of a single researcher and involve exploratory work where the priority is to be able to experiment and rapidly iterate, trying out different tools and approaches, and so on. As the project matures, the pipeline may get shared with and sometimes handed off to a wider team, particularly in the case of pipelines that are ultimately meant for use in production facilities, where the priorities are more focused on reliability and turnaround time. And of course, you don't want to have to re-implement the pipeline in a different workflow language at any point during the project. So you need a solution that can accommodate the full range. Key features that enable this include being readable, meaning collaborators can read and understand what the code does. Portable, meaning you can run the same code on a local machine, on an HPC, or on a cloud service with just a few configuration changes. Scalable, meaning that it's not going to fall over when you go from running 10 samples to 10,000. And reproducible, meaning that you can hand it off to a collaborator and they can readily run it on the same data and get the same results, which sounds like it would be a given, but you'd be surprised. The good news is that Nextflow satisfies all those requirements. It's open source and it runs just about anywhere. And I do want to call out the portability in particular, because having used other workflow systems myself before switching to Nextflow, I've been really impressed how truly portable Nextflow has proved to be, including in hybrid HPC cloud or multi-cloud setups. I also want to highlight that Nextflow provides native support for using a range of software packaging technologies, including Docker and Singularity containers, as well as Conda packages. And it offers built-in parallelization capabilities and data flow handling that are extremely powerful. All of this has contributed to making Nextflow the most rapidly growing pipelining framework today. But that's not all. With Nextflow, you also get NF-Core, which I mentioned earlier in my introduction. It's an independent open source project that brings together bioinformaticians across the world in a collaborative effort to develop high quality, community curated pipelines that you can use out of the box or customize to suit the needs of your particular project. There are over 100 pipelines like that, covering a wide range of omics domains. Or if you'd rather build from the ground up, you can take advantage of NF-Core's modularity principle, because most of the code in those pipelines is organized in modules and sub-workflows that you can use as building blocks to develop your own pipelines. There is so much to choose from. It can actually be a little overwhelming. Where do you start? And that's where I'd suggest you use a brand new tool that we recently released as a beta feature called Seqera AI, an AI assistant that uses large language models designed specifically to help you with your pipelining work. Now, I know we're all getting a bit of AI fatigue, but hear me out, because I'm going to talk about a couple of different things here that both have the potential to help you a lot. Number one is finding the right resources. As bioinformaticians, we all know we're supposed to use existing tools as much as possible rather than reinvent the wheel. But there is so much out there, and a lot of what you might find through a Google search or literature search is not actually fit for purpose. And AI can help you with that because it's a great tool for finding things and summarizing information efficiently. And the second thing I'll talk about is actual code generation, using the AI to develop your own pipelines more quickly, efficiently, and with high quality results. But first, let's talk about finding the right resources. The Seqera AI is available through our websites, so you can just go there and you can ask it to find a pipeline for the analysis type you're interested in. And the AI will recommend something that might be a good fit based on your description. Of course, the more detailed the description, the more likely it is that it will find you something suitable. Here I kept it fairly general and said, I want a pipeline for analyzing AttackSeek, so it recommended the AttackSeek pipeline from NF-Core. That was an easy one. But it also gave me a summary of what the pipeline does, including key features that are meant to help me determine quickly if I'm on the right track or not, and some basic instructions for how to run the pipeline, an example command, and some details about the required inputs, as well as the description of the pipeline's outputs, all of which is summarized from available documentation. Now, you might say, okay, but that's just what I'd get from Googling and reading the documentation, right? That's true, but because Seqera AI is a chat-based assistant, you can ask it to elaborate on anything you find in the summary. For example, you can ask it to explain what a particular process does, give you more details about input or output formats, and so on. You can even ask it to show you specific bits of code and explain what that code is doing. Personally, I find that very useful because it cuts down on a lot of time that I otherwise spend looking things up or asking around. And I can tell you, I ran the GHK support forum for 6 years or so, and a lot of questions we got could have easily been answered by an AI like this. So that's nice. But what about actual code generation, right? Let's talk about that. You might have seen the numbers around developers adopting code generation tools. It's everywhere, and for a good reason. For the most part, the capabilities of software like Copilot are really impressive. Even ChatGPT, which is super generic, does a really decent job of generating what I would call routine Python code. Like, for example, making an API call to GitHub to retrieve some usage statistics, something I've had to do myself. One time I was at Logan Airport in Boston. My boss DMs me on Slack to get the developer activity numbers ASAP, and I have 15 minutes before I have to board my plane. It's an international flight. Who knows if there's going to be Wi-Fi? So I pull up ChatGPT, 1 minute to put in the prompt and run it, 5 minutes of tweaking code, approximately, 2 minutes to run it, a couple of times, 3 minutes to write the email, which I should probably have used AI for that too. But I get the numbers, I get on the plane all right. And then we sit on the tarmac for an hour because of a technical issue. Anyway, my point is, it works really well for something like Python, ordinary routine Python. Can we use that for bioinformatics workflows? Well, the difficulty here is that generating scientific pipeline code is a much bigger lift than generating some basic Python code. Because with Python code, there's tons of it to train a model on, and a lot of what people are doing is the same thing over and over again. So that's really easy. In contrast, bioinformatics pipelines are harder because there's so much heterogeneity involved at every level. The level of the tools, the resource requirements, the complexity of the data sets, you have a lot of reproducibility issues, the list goes on. And there's not nearly as much training data as you'd want, ideally. But we had the great fortune of partnering with a couple of really clever guys who had been thinking about this specific problem for some time and had founded a tiny startup called Tiny Bio, appropriately enough. They've joined us, and we've been working together to make code generation actually work for bioinformatics pipelines. So, how do we make it work? First, you need a good training set, where good means well curated. You remember how I mentioned NF Core is a highly curated collection of workflows? That's actually a great resource for training an LLM because the NF Core pipelines apply high standards of code quality and they cover a wide array of use cases. So that's a great place to start from, which you wouldn't necessarily have an equivalent for with other workflow languages. And as a result, the Seqera AI is capable of generating valid Nextflow code for a wide variety of purposes. For example, I can ask it to write me a process, which is one step in my pipeline, that performs a particular analysis. Here is an example. I chose a variant calling step using a commonly used tool, GATK haplotype caller. And as you can see, the AI generated a complete process definition that includes all the relevant inputs, accessory files, and so on, as well as the correct parameter settings that correspond to the analysis mode I asked for. It even specifies an appropriate conda package for the tool. And if I wanted to, I could have it switch that out for a Docker container. This saves me a ton of time looking up documentation and typing out boilerplate code. Of course, I still have to check that it's actually correct because there's no way I'm going to treat this thing like a black box. So does it actually run and does it produce correct output? And this is where we get to my favorite part in all of this. Built-in testing. Anytime Seqera AI generates some code for you, it will also offer to test it, actually run it in a sandbox environment with real test data and everything. And it's a good thing too, because do you know what AIs have in common with human bioinformaticians? They get really confused by genome references. Here you see the AI grab test data where the reference genome is a subset of the whole genome, and the sequence data corresponds to a different chromosome compared to the subset that's in the reference file. And I've also seen a few cases where the AI ended up using different genome builds. You get a conflict there. So now, obviously, that's something we want to smooth out on our side, and that kind of goes hand in hand with improving test data collections in general. But what's really cool here is that the AI self-diagnoses. It's able to realize that there's a problem, identify what went wrong, and in most cases, it's actually able to self-correct, meaning fix the issue and run again to verify that the fix is correct. And let me tell you, after 10 years of doing online tech support of one form or another, the same cannot be said for every human. Now, here's another example where the AI runs into a couple of errors that have to do with input data being organized in a way that doesn't match the tool's expectations, where it progressively solves the problem just like a human analyst would, but much faster. And it eventually comes up with a solution that addresses the issue. So to be clear, sometimes the AI can take a few rounds to figure out a correct solution, but the beauty of it is that it doesn't actually require human intervention. So basically, it'll talk to itself for a few minutes until it gets the correct answer, which to me feels very relatable. So there's a lot more I could show you, but hopefully that gives you a taste of what Seqera AI can do. Now, it's currently in beta, and there's still plenty of work to do to get it to the point where you could confidently say, for example, write me an entire pipeline from scratch for something a little bit exotic, or convert this bash monstrosity that I inherited from another postdoc into beautiful Nextflow code. But it's already at the stage where it can potentially save you a lot of time writing boilerplate code. So please try it out and let us know what you think so we can keep improving it. That being said, even with AI doing a lot of the grunt work, you'll probably still need to do some good old-fashioned hands-on coding to get your pipeline just right. Which brings me to the other big thing I wanted to tell you about, which is the new version of our VS Code extension that substantially improves the quality of the Nextflow developer experience. If you're not familiar with VS Code, it's a very popular application for software development, an IDE, and we made an extension for it that unlocks a ton of functionality that makes it easier to write Nextflow code. Specifically, key features like syntax highlighting to display different kinds of code elements in different colors. Diagnostics tooling that tells you when you made a typo in a variable name or you forgot to provide a required input to a process. Hover hints that give you extra information about code elements. And auto-completion that suggests possible keywords or variable names as you type. And a number of other features, all of which you can read about in the documentation that describes the VS Code integration on the Nextflow documentation website. Now, I just want to call out specifically the hover hints and show you a few examples, just because I find them super useful to jog my memory and also save me a lot of time clicking and scrolling around to check what are the expected inputs for my processes and that sort of thing. So hover hints are really neat. I also want to point out this very discreet but tremendously useful feature, which is the preview DAG, meaning directed acyclic graph. This allows you to pull up a visual representation of the workflow graph. You can see the code is on the left for the workflow, and then on the right is the visual representation of this particular four-step workflow, showing you input parameters and all of that. When I started using Nextflow, there wasn't a way to easily generate this, so I was really thrilled when the team released it. It's really useful whether you're trying to understand what somebody else's pipeline does or trying to see if your own pipeline is kind of wired up correctly and so on. You can find the Nextflow extension in the VS Code extensions browser. Just search for Nextflow and click install to start taking advantage of this incredibly useful functionality. So if you're interested in learning more, visit our website at scalability.io or come ask us questions in our community forum at community.seqera.io. See you online.